Have you ever wondered how our bodies are able to make proteins that are essential for our survival and functioning? As we delve into this topic, I am going to explain how DNA synthesizes protein through a process of transcription and translation. Because I believe that understanding the process of how DNA makes protein is crucial in comprehending the complexity of life. The significance of making protein. Why is making protein even necessary? Well, we all know that the DNA contains all of the instructions for an organism to develop, survive, and reproduce. But these instructions has to be converted into messages to produce protein. Proteins is necessary to repair tissues, build muscles, fighting off infections. Those are only three examples I can think of with how why protein is necessary. However, protein is really important. It has many uses in our body. The process of making protein. This is just the basic protein, how protein is synthesized, like the process of how protein are synthesized. It starts with transcriptions, translation, and forming a polypeptide chain. I'm gonna go over that in the next slides. The central dogma. It all starts with the DNA. DNA stands for deoxyribonucleic acid, and it contains the genetic information that makes us us. And it also contains instructions for protein synthesis, as I mentioned in the previous slide. Then followed by RNA. The RNA contains a copy of the nucleotide from the DNA. The RNA is responsible for transporting the genetic information into ribosomes for protein synthesis. And lastly is the protein. This level is when the nucleotides of the RNA has been translated into amino acids to form different kinds of proteins. Proteins are found throughout the body. It is responsible for building muscles, repairing wounds, tissues, etc. Transcription of the RNA. This is the first step for protein synthesis. The DNA cannot get out of the nucleus. In order for the DNA's instructions to escape the nucleus, the DNA has to duplicate its genetic information. Therefore, certain nucleotides are duplicated to create a certain protein. The first step of duplicating the DNA has to split in two, and an enzyme called helicase right here is responsible for splitting the DNA in half. And it does this by unwinding the double helix and by breaking the hydrogen bonds between the base pairs. So what it means is that the helicase just splits this double helix right here. And these like colorful lines are the nitrogenous base. And it splits it destroys the hydrogen bonds in order for the double helix to split in half. But you may be wondering, how does the helicase know where to start cutting? Well, the helicase knows where to start cutting in an area called the promoter. And each promoter will be different based on the protein that the DNA is trying to synthesize. So a promoter could be right here. This is where the helicase could start. It could be right here. It also could be right here. It all depends on what kind of protein the DNA is trying to synthesize. So once the DNA is split in two halves, the first half gets healed or zipped up in order to repair the double helix, and it help like an enzyme that helps this process is called the DNA polymerase. And it does this by adding complementary nucleotides to the existing strand, while the other halves becomes the RNA. The RNA is single-stranded. It contains uracil instead of thymine, and the DNA has thymine. The base pairs for thymine is for DNA is adenine A always attaches to thymine T, and cytosine C always attaches to guanine G. However, in the RNA, instead of using thymine, the thymine is replaced with uracil. The RNA is then transported as mRNA, called the messenger RNA, into the ribosomes on the cytoplasms for translation. Once the mRNA attached to the ribosomes, it will attach to the lower subunit of the ribosomes. This is the lower subunit, and the orientation of the mRNA is kind of like upside down. And each lines right here are called amino acids. These are all amino acids, and every group of 3 amino acids are called the codon. And this big red thing right here is called the large subunit of the ribosomes. And the large subunit ribosomes has 3 attachment sites. It's the A sites, P sites, and E sites. You can see in this thing right here too. This is the A, P, E. And this yellow thing right here is called the tRNA, called the transfer RNA. It first attaches itself to the A sites with its corresponding nucleotides. It is called an anticodon, right? The tRNA lands itself to the A sites of the large subunit of the ribosomes with its corresponding anticodon that matches the mRNA's codon. The tRNA then shifts to the P site, which is right here, while simultaneously another tRNA attaches itself to the A site. When the tRNA attaches to the E site, this is called the exit site. And this is when the tRNA exits out of the ribosomes. However, as it exits the tRNA leaves its amino acids, forming a peptide bond. I couldn't really show a better picture for it, but the amino acids will be right here, and it will attach to each tRNA. And as it leaves these amino acids with it, and then eventually, as the mRNA reaches to the 5' cap, the protein is like made. It will form like a whole polypeptide chain, and this polypeptide chain is what protein is. So in conclusion, to summarize everything mentioned in this topic, the DNA, short for deoxyribonucleic acid, contains the genetic information necessary for protein synthesis and is the fundamental basis of life. 2. The process of making protein from DNA is called protein synthesis. It involves 2 steps: transcription, the creation of RNA, and translation. It is the process of translating the nucleotides of the RNA into amino acids to synthesize protein. 3. The group of 3 nucleotides of the mRNA is called a codon. The tRNA matches its codon with its anticodon. And once the polypeptide chain is formed, the protein is synthesized. In conclusion, by understanding how DNA makes protein is essential in comprehending the molecular basis of life. Proteins are important because it helps build muscle, repair wounds and tissues, fight off infections, etc. And it is crazy to think that the DNA holds all of these instructions for making proteins, right? So that is it for my presentation, and these are my references. Thank you.